At this moment, it is my great pleasure to introduce to you our resource speaker for today, who is going to talk about the importance of advances in biological research. Our speaker for today is a graduate of Bachelor of Science in Biology at Mindanao State University, Main Campus. He finished his Master of Science in Microbiology, minor in Molecular Biology, and Biotechnology in University of the Philippines, Los Baños, year 2019. He was a thesis dissertation grant awardee in Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development, Department of Science and Technology from August 2017 to December 2018. He was awarded as the best paper in biological sciences category in 14th Annual Scientific Meeting, National Academy of Science and Technology, Philippines. He was currently working as an instructor at Cavite State University, Don Severino de las Alas campus. He conducted a research entitled Assessment of the Effect of Combining Biochar, Biofertilizer, and Vermicompost on Soil Quality and Crop Production, implemented from 2015 to 2019. Ladies and gentlemen, let us all welcome Mr. Angelbert Damian Cortes. A round of virtual applause.
Right. Thank you very much, Anne, for that generous introduction. Thank you very much also for CSU or CARSU for inviting me to talk, especially Joshin. Thank you for personally inviting me here. And Joshin is a good friend of mine since college. And right, so I hope that our viewers today are students actually, from different universities. And I also have here colleagues from our university. Right, so let me now share my slide to start my presentation. Right. Okay, so of course, I would also like to extend my gratitude to the chairperson of the Department of Biology of CSU, Dr. His name is Demetilio. Am I right? Right. So thank you very much, sir, for your support in this webinar. And of course, the students from genetics and cell molecular biology. This is a very good and timely webinar for you guys so that you will be also engaged in doing research soon. All right, so my talk for today is, okay, focusing on my research about the application of biochar and arbuscular mycorrhizal fungi as agricultural options for better crop production. So as you can see in the background, this is the specific crop that I am focused with. So I am from the Department of Biological Sciences of Cavite State University. Is unstable. All right. Now, my talk for today includes, or the contents of my presentation includes the background of biochar and arbuscular mycorrhizal fungi. Here, I'm going to discuss to you the nature and importance of biochar and AMF in agricultural biotechnology. Then also, I will share to you the research findings based on my research na sa thesis ko sa MS. I'm going to discuss to you or share to you some of the research findings on the application of these treatments on cacao plants. And also lastly, I will also share to you the application of these treatments on other crops. All right, so let me start my presentation by sharing to you the definition of biochar and arbuscular mycorrhizal fungi. When you hear the word biochar, it's somewhat like in Bisaya, we usually say char, right? So the biochar is actually a short term for biological charcoal, but the difference is that in biochar, this is thermally decomposed carbon-rich product, which is obtained through pyrolysis of biomass. Biomass can be manure, wood, or leaves, or any kind of biomass can be converted into a charcoal. But the difference is that you are not applying oxygen, so it means that during the process, the system should be free from oxygen so that the product is not burned. Instead, it's the heat that will decompose the product so that the nutrients will stay within this biomass. So that's why we call that biochar. And again, it's a pyrolyzed product. So it's rich in carbon dioxide. And during the process, you emit oxygen so that hindi siya masusunog. Now, we have also here the arbuscular mycorrhizal fungi. If you can see this figure, this is a cross-section of a root part of the plant. And in this part, you can see, nakita niyo ang cursor? Nakita lang ba ang cursor? Hello?
Yes,
Yes,
sir
Bong.
po.
Yes, Bong.
Okay,
Yes,
okay.
sir.
Thank you. So here is the part of the non-mycorrhizal. It means that wala siyang infection of the AMF. This part is the ectomycorrhizal fungi. Means nasa labas lang siya ng root part. And this is specifically for angiosperm, okay, for the flowering plants. This one is for the gymnosperms. But my fungi that we use now, or I used in this study, is specifically the arbuscular mycorrhizal fungi. So these fungi extend its mycelium inside the cortical cells of the vesicles, forming an arbuscule. That's why it's called arbuscular mycorrhizal fungi. So for your information, these fungi constitute a group of root obligate biotrophs that exchange mutual benefits with about 80% of terrestrial plants. So basically, you can find them in the rhizosphere or even in the root system of majority of the terrestrial plants. Now, it is interesting to share to you that this kind of product, or let's say biomass or powerless product, biochar, is very important, especially for agricultural purposes. So here, example, we use bamboo biochar. There are several biomass that you can use, you know, to convert it into biochar, but here we use bamboo. Now, why bamboo? Okay, as we know that bamboo products or those waste products are just being disposed by bamboo related industries. So these kind of activity might cause a release of carbon dioxide in the atmosphere, and it can cause additional greenhouse gas emissions, and as we know, it can lead to climate change. And it is already evident right now that we are experiencing severe climate change. So in order to minimize this kind of activity, we utilize this product and make it useful for environmental purposes and also agricultural purposes. So the biochar is powdered, before it is amended to the soil as a conditioner. So I just want to emphasize that this product is not fertilizer. Instead, it is a soil conditioner. So this one has a role in plant productivity by improving water holding capacity. So it means that when the plant is exposed to drought, this plant can survive with the help of biochar because the biochar holds the water so that this water will be available for plants consumption. We also have here, it improves soil fertility because the nutrients itself, biochar itself, is very rich in nutrients. So basically, madaming nutrients ang provide ng biochar sa plant. And since it holds the water, it also holds the nutrients for plants' nutrition. Now, since it improves the water holding capacity and soil fertility, the turnover will be a good crop productivity because of the provision of nutrients now for plants' growth and development. And one of the promising effect of this biochar is it actually mitigates carbon dioxide emissions. So it enhances carbon sequestration capacity or ability. So pag sinabing carbon sequestration, it will not allow the reaction of carbon to oxygen to form carbon dioxide. Instead, it sequesters carbon. So it holds carbon, therefore carbon dioxide emission is mitigated. Now, the last one is the very interesting part when I read these references about the role of biochar in microorganisms. Since my major is microbiology, I was captured by this important importance that it serves as a refuge for colonizing microorganisms. So that's why my research really focusing on this. So how the biochar cater the microbial communities in the soil or rhizosphere specifically. Now, there are several studies that couple this biochar with biofertilizer, and this is the arbuscular mycorrhizal fungi. So arbuscular mycorrhizal fungi, or AMF, is again a natural biofertilizer which shares association with plants. The plants provide carbohydrates for the consumption of the fungi, and in return, the fungi provides nutrients. All right, so this figure shows the interaction of the fungi. We have here an example, Gigaspora spore. So they are spores actually in nature. So the spore extend its mycelium into the root vesicles, specifically in the cortical cells of the vesicles of the root system. So it forms an arbuscule, so therefore it's called arbuscular mycorrhizal fungi. And if you can see in this figure, you have here processes, nitrogen fixation and phosphate stabilisation, wherein these are driven by microorganisms such as the nitrogen fixing bacteria and the phosphate solubilizing bacteria, respectively. So they have this association with plant growth promoting rhizobacteria. So this ammonia and sorry, so this ammonia and phosphorus sorry, is being utilized by the plant phase, and then sorry for that. All right, so the hyphae class will serve, or not class, sorry. Hyphae will serve as a channel for the nutrients to be uptaken into the root part. So basically, sila yung magiging channel para ma-uptake yung nutrients from the soil towards the plant through the root system. You can see here that there are several benefits of these fungi. First is it has it inhibits the pathogen by forming biofilm. The biofilm protects the root system, therefore the pathogens cannot inhibit the root system. And then it also improves nutrient uptake and also hyphal growth stimulation. Now, when it comes to the spores that we use, or fungi that we use in this study, we use 12 species or species of AMF belonging to the genera Glomus, Lycospora, Apalospora, and Anthropospora. So here, these fungi improve water acquisition. It also improves nutrient acquisition. So if you can see kanina, biochar improves water holding capacity and nutrient availability in the soil. So these fungi will improve the uptake of the nutrients and also improve the uptake of water. So it really has a good or beneficial effect to the plant for the plant's productivity. So it also protects the plant against pathogens. As I've said in this figure, it forms a biofilm that protects the plant from pathogens. And lastly, these fungi is also beneficial, especially when the plants are experiencing drought and also heavy metal contamination. So I just want to share that these fungi, I was involved in research in UPLB, specifically in biotech now. We conducted a research by utilizing these fungi in improving the growth of the crops in mining areas. And fortunately, we were able to revive the dead soil alive. That's our tagline now, because this fungi is really important for bioremediation purposes. So the interaction of biochar and mycorrhizal fungi is a current interest due to their contribution to sustainable plant production and ecosystem restoration. So based on the importance that I've mentioned kanina, you can really see how important it is to explore the biochar and arbuscular mycorrhizal fungi as agricultural options to improve crop production. So here we apply the treatments in the cacao plant. According to the 2017-2022 Philippine Cacao Industry Roadmap, cacao is an important crop for chocolate and cocoa production, and this crop is now in high demand worldwide, yet faces big challenges, especially in cultivation. That's why farmers now often need support to make their farm more productive and profitable. And according to the Bureau of Planted Industry of the Department of Agriculture, cacao production or the global demand is expected to reach between 4.7 to 5 million metric tons. So imagine. And the consumption in the Philippines is 50,000 metric tons, but our local supply is only about 10,000 metric tons, making the Philippines a net importer of these products. So the goal of these 2017-2022 Philippine Cacao Roadmap is to improve the production of cacao up to 100,000 metric tons annually. And this research is actually a contribution to this roadmap, since we were able to demonstrate the effectivity of the biochar and AMF in improving the growth of cacao in acidic soil environment. Why cacao? So this question is a very common, what they call this, primary question being asked by those who have reservations now about the potential of the cacao industry. So cacao is important because it requires small monetary investment or startup capital. And if you have farms there, you can observe that majority of the farmers, like 90% of farmers, utilize this field and make it useful to produce the cacao crabs. So it requires small monetary investment actually, and that's why mas maraming farmers they prefer utilizing their farm para makapag-produce ng cacao. So the cacao is also suitable intercrop for coconut and banana, which is the primary products of the Philippines. And cacao is a 2 week harvest interval. It requires only 2 week harvest interval, and the gestation period is about 18 months. So when you cultivate this crop after 18 months, you will already have the produce. Now, early return investment is also important factor as to why there are farmers who are still interested in the cacao. No? So high profitability of the product is also a good consideration, ensuring good income augmentation. And lastly, cacao industry is market driven because cacao has no substitute product. So if you love chocolate, then those chocolate are actually part of, derived from cacao. So basically, cacao may have significantly contribute to poverty alleviation and also inclusive growth through livelihood and job generation. Now, I just want to share that cacao has huge contributions in the industry by producing these cocoa-based products. So here we have the wet cacao beans, cacao seedlings, cacao leaves, tablea, which is very common to us, especially when we make chocolate or even champurrado. And then we have cocoa powder, which is used for cake production, cocoa butter, chocolate, which is the primary product derived from cacao, and then the chocolate confectionery means that different varieties of chocolates. Now, here I'm going to share to you how we tested the biochar into the cacao crabs. So we have actually this greenhouse wherein our crabs were set up, and as you can see, this is I think this is a 4 month old cacao seedlings. So we have separated them based on our experimental design. We have treatments, and it's elevated because it prevents to avoid exposure to flooding in case of flooding. And the environment is also controlled. We put nets, and the sunlight penetration is also minimized. So the components, we use soil sand substrate and we use acidic soil to make sure or to have a significance. Kasi in the Philippines, there are places no na sobrang acidic ng soil. So therefore, mahirap mag-grow yung mga plants. So therefore, we use this acidic soil to explore the effectivity of the treatments in improving the crop, even though they are exposed to acidic environment. Bubble biochar amendment, so we use different concentrations of biochar here, 7.5, 3.75, 7.5, and 15%. And of course, AMF inoculation. My laboratory, I mean, the laboratory of my boss, which is a scientist in UP, produced this product, and we utilize those species of AMF, it's cacao. And then, of course, the variety that we use is cacao UF18. Now, for our treatments, we have major treatments, control those with AMF alone, and then mixture of biochar and soil, and then biochar mixture plus AMF. So we actually have a lot of treatments, but these are just major treatments. All right. Now, for the data collection, I just want to share that we get data from the cacao growth itself. So you can take note of this and you can apply this when you are planning to do research also in this kind of field now. So you can get the cacao growth. Here we measured the height, also the stem diameter, the yung weight ng crab, and so on. So we also get the data for soil chemical properties like the NPK, CEC, OM from the soil. And my research is really focused into biological properties since my major is microbiology. So I focused on analysis of the bacterial population count, specifically those that are plant group promoting rhizobacteria. We have the nitrogen fixing bacteria and the phosphate solubilizing bacteria. We also analyzed the infection of the AMF sa root and also counted the spores of AMF in the soil. And also, we perform the microbiome profiling using next generation sequencing. So later on, I will discuss to you or show you the result of this research. Now, why it is important to analyze the microorganisms in the rhizosphere? As we know that microorganisms are natural partners of plant. So if you can see here, it's very common class in the environment that these microorganisms interact with plant. And that's why in microbiology, we say that in biology, you are taught that 80% of the population of these microorganisms are beneficial, 3% lang ang pathogenic, and the rest are opportunistic. So you can see that processes in the environment, they are driven by important microorganisms. So some of the microorganisms are deleterious to the plant, like those pathogens, like Alternia, for example, they cause infections of root, the Fusarium, but there are also organisms that have neutral effects. But the important part of this is you have to focus with these beneficial microorganisms like the mycorrhizae, infecting bacteria, and then we have those microorganisms that increase the uptake of the carbon, nitrogen, phosphorus, and so on. So we call them as beneficial microorganisms. Specifically, we call them plant growth promoting rhizobacteria. That's why get interested ako with these microorganisms. Now, in this paper, I just want to share that we were able to demonstrate the population or the effect of the treatments on the population of plant growth promoting rhizobacteria and also the plant uptake. No? So we in this table, I will show you that the soil that we use is very poor when it comes to its nutrient status, and as you can see, the pH is very, it's very, it's acidic. No? So basically, it's very poor when it comes to its nutrient status. But the biochar that we use is very rich, when it comes to nutrient status, and the pH is alkaline. So if you combine this, there's a possibility that the pH will increase and the acidic soil will be supplemented with biochar nutrients. That's why they are called as conditioners. So here we demonstrated an increased uptake of the nitrogen and phosphorus into the plants. So we concluded that AMF improved the uptake of the cacao about 60 to 90% for nitrogen and phosphorus respectively. So it's promising, right? And then N uptake was highest in those plants that are treated with AMF and 15% BB. So overall talaga, the AMF and 15% BB combination has good contribution in improving the growth of the plants. And then the uptake was generally improved by biochar. All right. So when it comes to the population, we demonstrated that the biochar at 15% improved the population of the culturable NFB and PSB. So as I've mentioned, the role of biochar serves as the refuge for colonizing microorganisms. And in fact, we demonstrated that these microorganisms, specifically the plant growth promoting rhizobacteria, NFB and PSB, was stimulated when biochar was treated. So here I show, I'm sharing all the pictures that I have documented when I had my thesis. So you can see here the population of colonies, no? Or the colonies on the medium. So we use here, I use nitrogen-free maleate mediums. It means that walang nitrogen in this medium. So the microorganisms that grow here are nitrogen fixers because they only fix atmospheric nitrogen from the atmosphere, from the air. So yeah, so that's why they can grow on the medium. Here naman are the phosphate stabilizing bacteria. So as you can see that the colonies, the colonies have clearing around them. So it indicates phosphate stabilization. So I consider this as phosphate stabilizers. Now, in this paper, I'm going to share also the effect of the treatments on the cacao growth itself. So as you can see, we compared the effect of the treatments on the cacao plant when it's grown in sterilized and sterilized soil. So why we compared it with the sterilized and sterilized soil? So as we know that we are using microorganisms and microorganisms are present everywhere. So we tried to elucidate whether the AMF will be affected by the indigenous microorganisms or wala silang effect, or there will be a mutual relationship. So let's see. Right. So here we demonstrated an increase in the growth of crops. So specifically, AMF increased post-growth traits of cacao plants. When it comes to growth traits, the plant height, the dry weight of the leaf, the stem, and also the roots. So basically, overall, nag-increase yung growth niya. And then, of course, when it comes to biochar amendment, 15% gave the best plant growth, regardless of soil sterilization. And when it comes to the dry weight, AMF and 15% BB, so the combination of AMF plus 15% BB provided the heaviest plant no, among the treatments. And lastly, when it comes to the diameter and the height of the plant, biochar and AMF increase these parameters. So you can see that in this paper, we demonstrated a very good effects of the biochar and AMF in improving the growth of cacao seedlings. So here, this figure shows the increment values. So we actually measured the height and stem diameter from 0 to 15 months. All right. So regularly yun minimeasure namin. And you can see in the figure that 15% BB consistently improve the stem diameter and the height of a plant. Here in this table, although it's okay that hindi niyo siya masyado ma-follow it because it's very complex data, but I'm just showing here some of the results that we have, promising result. Let's focus on the total dry weight na lang. As you can see that statistically, with the presence of AMF, okay, it is significantly different from those without AMF. And when it comes to presence of biochar, 15% BB showed a good effect on the total plant dry weight. So nag-increase yung dry weight niya. So it means that, okay, madaming nutrients. Pag sa tao to, malusog ka because you have provided with enough nutrients. Ganun yung effect ng biochar sa plant, specifically sa cacao. Okay, in this table naman, this data shows naman the mycorrhizal root infection. As you can see that, again, it's quite complex, pero let's focus on the general mean values. As you can see that 15% BB showed highest infection rate consistently in sterilized and unsterilized soil. When it comes to the combination, the 50% BB and AMF also showed highest infection rate, both in sterilized and sterilized soil. Now, when it comes to spore count, this one, we just counted the spores in the rhizosphere. Still the same results. Biochar improves the spore count in the rhizosphere. So this figure shows the uninfected root part of the cacao. Wala kang nakikitang arbuscular mycorrhizal fungi, diba? So it's plainly a root. Here is the figure showing the root containing the spores infecting the root system. And this one shows the spores that are collected from the soil. Okay, so we counted them, the population. Now, when it comes to the soil physical chemical properties, generally, again, I'm going to summarize the lang. Generally, biochar, 15% BB biochar improve the majority of the chemical properties. For example, here, the phosphorus, I just look at letter A. Letter A indicates highest value. So here, A, in unsterilized soil, it is in 15% BB. In unsterilized soil, still in 15% BB, regardless of AMF inoculation. Also here, 15% and so on. So basically, majority of the chemical properties was improved by biochar. And again, if we get back to the introduction part, I mentioned about acquisition of nutrients. So it holds the nutrients within the soil environment, and this provides an information supporting that kind of knowledge that biochar really holds nutrients for plants' nutrition. But the page here doesn't really have any changes, pero you can see there is a slight increase, pero not significant. Okay, so again, biochar is evident in improving the nutrient availability for plant, and chemical properties was also or were also improved when biochar was amended. AMF, medyo wala siyang effect, pero biochar has huge effect in chemical properties. All right, so in this paper, which is published last December 2020, I also demonstrated the role of the isolates that I have isolated from the rhizosphere, especially from those counted population known doon sa isang paper. So here, I analyzed the ability of the isolates to fix atmospheric nitrogen by performing experiments to prove that there are really diazotropic bacteria. Diazotropic bacteria is another word for nitrogen fixing bacteria. So basically, they fix atmospheric nitrogen. So as we know that biochar serves as a refuge for colonizing microbes. So it affects the mycorrhization, helping bacteria. That's why my research here focused on the characterization of these plant growth promoting bacteria. So I performed acetylene reduction. So this is the standard assay if you want to test for the nitrogen fixing ability of your isolates. You can do, all right. Okay, so acetylene reduction will reduce acetylene to ethylene gas. And this activity is driven by the nitrogenase, which is present in these nitrogen fixing bacteria. We also perform phosphate stabilisation, and of course, we identify the isolates using 16S RNA gene analysis. So this one is the assay for acetylene reduction. So I stubbed the culture in the semi-solid medium. It's very small tube para maximize yung growth. And if you can see, the original color of the medium is green. And once nagkaroon siya ng blue color, it means that there is growth now of microorganisms. So I incubated the culture for 24 hours and replaced these cotton plugs with the rubber septum. So after that, I replaced it so that the gas will not leak. No, kasi I will test gas here. So after that, I removed 2 ml of air and replaced it with 2 ml of acetylene gas. So I used a sterile syringe in order to maintain uh, you know, the aseptic technique or aseptic way of culturing the microorganisms and also to prevent contamination of the sample. And after that, the culture is further incubated for 24 hours and we tested it. The ethylene produced was tested using gas chromatography at Biotech. So later on, I will show you the result. Now for the phosphate solubilization, I also performed assay, which is adapted from one of the papers that I used. So here, this is not disc, yung paper disc, but I used broth culture. I just put 2 UL of broth culture in triplicates and allow it to be incubated for 9 days for at 30 degrees Celsius. After 9 days, these are the results. As you can see, itong culture na to, walang clearing around the colony. This one is the control, the Pseudomonas aeruginosa. It has the ability to solubilize phosphate. And this one is a promising isolate. I was shocked when I observed this because mas mataas yung clearing niya or mas malaki yung clearing niya than the control. Right? So this clearing indicates phosphate solubilization of this culture. And again, this media we use is a Pagpanuhid medium. This is a good medium to test whether your isolate has the ability to solubilize phosphate. This one naman, after we tested the ability to fix atmospheric nitrogen and solubilize phosphate, I subjected them to molecular identification by extracting their DNA. So this figure, although the marker is not so bad, right? Pero just to emphasize the DNA here, I got DNA from my isolates, although some of the bands are very faint. And I also observed bonds here that I suspect these are plasmids, and these one are just residues. So this gel shows the DNA extracted from my isolates. And then after that, I performed PCR amplification targeting the 16S RNA gene of the bacteria. So if you are working with bacteria, 16S RNA gene is a good gene or marker for you to identify them. Okay, so these are the products, although it's quite not good. It's not good actually, but I was still able to identify them using this products. So as you can see that the DNA bonds aligned with the marker. We have the 16 or 1,600 base pair or 1.6 kilo base pair. So the size of the 16S RNA is about 5,900 base pair. So I would say that these are my targets because it actually aligns with the marker 1.6 kb. And then I sent samples to Malaysia and sequenced it. And then after 3 days, they sent me the sequence. Now, I have here a figure or the figures showing the possible result of your sequence once you identify your bacteria. It doesn't mean that you have the sequence, meron ka ng result at masaya ka na. There's a tendency that you will get two results. First is you will get a good quality sequence. By the way, I use a sequence scanner here to scan the sequence and see the chromatogram of my sequence. So as you can see that it's very nice, it's neat, and each peak represents base. Like, di ba, sa biology, meron tayong adenine, guanine, cytosine, thymine, and it is represented by peak or peaks. So this is a good sequence, and you can say that you will get really a good data from here. But this figure shows a bad quality sequence because you can see a lot of noises in the sequence. It means that your sample maybe is contaminated with two or more species of microorganism. And before you extract your DNA, you have to make sure that your culture or bacteria, your bacterial culture is pure. You can um, uh, you can do that by doing gram staining. It's a good way to determine whether your culture is pure or not, because you can see the same color, the same shape of the size. Since we are talking about bacteria, hindi nyo man sila nakikita with your naked eye. All right. After that, I further analyzed the sequence. Blast N is available when you Google it. Blast N, lalabas ito. So this is a good free access website wherein you can scan your sequence. So just copy the sequence here and click the mga options na necessary for your analysis, and it will give you possible candidate organisms of your sequence. Pero hindi ibig sabihin na sa first line, siya na yung candidate mo because more or less, ang daming representative ng microorganisms mo. So you have to download all the sequences that have 97%. It's not visible here, but you have to download all the sequences with 97% identity and then combine it with your sequence and then align it using Mega 7. So I'm using Mega 7, although right now meron tayong mga Mega 10 or meron ng Mega 10 pero my laptop is not compatible with that. So you can align the sequence using Mega 7 and you can generate a phylogenetic tree. So paano ko na-identify yung organism ko? So for example, NFP14. When the NFP14 clustered with this sequence, so most likely your NFP14 is Ralstonia manitolitica. So that's the identity of this NFP14 kasi doon siya nag-cluster. So that's how I identify the organism. So this is a table showing the results of my experiment in that paper. So I found out that all the NFB isolates performed ethylene production, so indicating that they are nitrogen fixing bacteria. And 11 isolates out of 12 showed phosphate stabilisation. And also, based on my analysis or molecular identification, I found out or I analyzed, or let's say my results showed that all the isolates belong to the genera Bacillus, Burkholderia, Rhizobium, and Ralstonia. These genera, if you read articles, these are known as nitrogen fixing microorganisms. So basically, it is supported by evidence. And lastly, this is the last part of my research, focusing on the taxonomic microbiome profile. Kung kanina, I focused on specific group, but here I focused on the community. You have to take note that 99% of the population of microorganisms in the soil is not culturable. So therefore, you need to apply molecular technique. And the very good one is the next generation sequencing approach, because here you can determine the possible occurrence of microorganisms kung sino-sino sila within that soil. So in this paper, I discussed, this paper was recently published last March 2021, okay, at Philippine Agricultural Scientists. So here, the metagenomic analysis of the V3V4 region of the 16S RNA gene in the rhizosphere revealed that the sample's diversity, pag sinabing sample's diversity, yung microorganisms sa sample ko ay na-reduced by the treatment. But hindi ako nabahala. I was actually um, worried when I found out that why is it that the diversity decreased when the treated is applied. But when I tried to analyze and compare it with the growth of the plant, nag-improve yung plant growth. So therefore, there's something wrong within that community na na-stimulate maybe yung mga beneficial at na-reduce yung mga deleterious microorganisms. All right, so for the method, if you're wondering how next generation works and our next generation sequencing works, maybe the faculty members of CSU and the students can explore this experiment because this is a uh, current research now when you want to identify the community of microorganisms in the soil. I performed community DNA extraction. And I just want to share that I experienced several failures when I did this because my sample is recalcitrant. It's not cooperating. So I made several modifications, such as additional of 40 milligrams of skim milk from reference, and then I applied several washing. And true enough, I come up with the result, but the DNA is very faint. Okay, I was worried because the sequencing services required high quality DNA, high concentration. The quality of my DNA falls within the range 1.8 to 2, but the quantity is less than 10 nanogram microliter. So here I was still hoping to get more DNA, so I extracted, but my sample is already old kasi nasa ref lang siya at negative 80, so degraded na yung DNA ko. So my kit was already used up, so ubus na yung kit. So therefore, I just sent the samples, bahala na. And true enough, the Asia Gel, or the first base in Malaysia, performed PCR amplification of the V3V4 region of the 16S RNA gene. And I was happy when they sent me the result because they were able to amplify the region. And this indicates that my sample passed the quality control. So again, it doesn't really mean that if you get DNA, you are happy because you will still wait whether your DNA can pass the quality control. So this figure, I was happy when I received this because, hi, salamat, at nakapasa yung sample ko for the quality control for the next generation sequencing. So further, they analyzed or they performed library preparation and then the sequencing using this Illumina Music platform. There are several sequencing technologies that are available, but I use the Music, which is appropriate for my study. And lastly, they perform bioinformatics analysis and they analyze the alpha and beta diversity. This kind of research is quite easy if you get high quality DNA and mataas na concentration ng DNA. Kasi all the majority of the experiments will be performed by the service provider. Okay, so ang babalik sa iyo ay data, so which is yung i-represent ko dito ngayon. So here is the alpha diversity. When it comes to alpha diversity, it means that this is a diversity within the sample. Okay, so here if you can see that the number of OTUs means operational taxonomic units. Ito yung nag-represent per genus ng microorganism present sa sample. So therefore, there are at least 4,096 OTUs present in the control. But if you can see that in the treated samples, bumababa yung OTU numbers. All right. So here, the Chow1, a numbered observed species in this table, shows that the treatments reduced species richness of acidic cacao rhizosphere. So bumababa yung richness ng microorganism. And also based on the Shannon and Simpson's indices, microorganisms in untreated soil was more diverse as compared to the treated soil samples. So I was really worried with this result because in the references, in most of the references, they provided an information that the species diversity and richness should increase upon treatment. But in this research, baliktad yung result ko. So, but I was happy again because when I tried to correlate it with the plant sample, the growth of the plant, reverse yung effect niya. Nag-grow yung plant significantly sa treatments kahit na bumababa yung species diversity niya. So here it is a refraction curve. It shows the cumulative number of species represented by OTUs. As you can see that as the sequencing increases, okay, hindi pa siya actually nag-plateau, pero makikita natin A1 is represented by the control. Mas mataas yung species richness niya as compared to the treated ones. Now, when it comes to the microbial community structure, which is represented by the beta diversity, meaning diversity across the samples. So I compared the diversity across the samples, and this is the data that I gathered. So here, you can see that the this figure represent the phylum. Pag sinabing sa microbiology niyo, may tinatawag pa yung mga phylum, so, or phyla, sorry. Each phylum is represented by different groups of organisms. For example, proteobacteria. So here, the proteobacteria class, this phylum is represented by plant growth promoting rhizobacteria. And true enough that based on my result, di ba bumababa yung species richness kanina and diversity, tumaas yung relative abundance nila. So they are stimulated, and these are beneficial microorganisms. And this is somehow contributing to the improvement of the growth now when I reapply the treatments doon sa cacao. And this one figure showing the classes, okay, represented by different classes under this phylum. Specifically, the gamma proteobacteria represented by microorganisms that are health indicators of the plant. Ay na-stimulate sila with the treatments. And of course, at 15% BB and AMF, na-stimulate ang alpha proteobacteria. Alpha proteobacteria includes bacteria that are plant growth promoting rhizobacteria. So it has really, this is a very good result now that I have gathered. And this conclude that the treatments have really a good effect doon sa plant because it stimulates the microorganisms present in the rhizosphere. Okay, this table naman shows the uh, genera naman. Okay, mga genera. So you can see that specifically rhodoplanes was improved when AMF plus 15% BB was applied. Rhodoplane class is a nitrogen fixing bacteria, and it's under alpha proteobacteria. We also have here Bacterialis, although it's still unclassified. It means that this is a novel species, but it is, I don't know, I was able to culture it, but based on molecular identification, it is still unclassified. So specifically this one, 13.3. So there is a huge increase now from 1.2 to 13.3% relative abundance upon biochar amendment. So basically, this biochar improved this Acidobacterialis. We also have here the Chromatialis, still unclassified. It also improved significantly when biochar was amended. Chromatialis class, these are plant health indicators. It means that if they're present, the plant is somewhat like healthy because it protects the plant from pathogens. And lastly, we have the Santamanidalis, still unclassified genus, and it was also improved with biochar. So this figure shows the PICOA or the principal coordinate analysis. As you can see that those uh, shapes, triangle, square, reverse triangle, and then circle, it is represented by my sample. And if they're separated for each quadrant, it means that itong community na to is unique from this, is unique from this, is unique from this. So this indicates that the microbial community structure is unique across the samples. Okay, and it has a huge contribution, especially when it comes to its effect on the growth of the crops. This one man shows the weighted unifrac. I'm sorry if you're overwhelmed with the figures, but if you can see that based on the community, the biochar, AMF, and those combined with biochar, so both, as you can see, it clustered together. So there is somewhat like a closer relationship, especially those with AMF inoculation. And this A1 is the control without treatment. It clustered away from the two treatments. But here, A3, which is plainly applied with biochar alone, it has a, what they call this, it clustered away from the rest of the samples. So it means that biochar has actually a clear or contribute a clear shift in the rhizosphere microbial community structure. Okay, especially when the soil when the crop is grown in acidic soil. So this Venn diagram shows yung OTUs lang which is unique and then common to the samples. And it only shares about 4.9%. No? This 685, ito yung mga genera that are shared across the samples. All right, for the key findings of this research, there are several factors that affecting the ecological shifts in the relative abundance of the taxa. Okay, first is those organisms that are enriched, okay, are maybe those are stimulated. They're actually enriched, no, by the nutrients that are provided by biochar. And those naman that are reduced, somehow biochar is toxic to them. That's why their relative abundance decreased upon application. And then we have here the changes in the properties of biochar amended soil. As you can see that the the it's not just in the microorganism but also in the chemical properties and the growth itself. It has really a change now due to the amendment of biochar. And lastly, the plant AMF symbiosis also has contribution to the microbial community shifting. Okay, and then the stimulation of these plant growth promoting microorganism and plant growth through agricultural options pose important indications in agricultural development. That's why we recommend that farmers can adopt this option because if you can see, we demonstrated a very good effect now of these treatments on the growth of the crops. And not just in the growth, we also analyze the effects on the soil and even in the microorganisms, which are the drivers of nutrient availability in the soil. All right. The last part of this lecture is I'm just going to present to you an overview of the application or the research development of these of the biochar and AMF on other crops. So this paper cited, or it was recently published in January, cited all the researchers that conducted the application of biochar and AMF on the crops. So here, the combined application, as they suggested, of biochar and AMF could open promising way for soil rejuvenation. Di ba pag sabing rejuvenation, there is a sumobra na ako isang oras na. Rejuvenation, like for the skincare products, you're applying it so that your skin will rejuvenate. So just like the soil, you're also putting this kind of application because you want to rejuvenate the soil. So you want to condition the soil and improve their nutrients. So these play crucial roles in environmental management. So to give you a summary, again, the combination of biochar and AMF enhances the uptake of the nutrients, help in nutrient recycling, reduces carbon dioxide emission because it has the ability to sequester carbon, provide habitat for microorganisms, helpful in remediating heavy metals, and of course, the turnover will be improve the crop yield and productivity. This table shows the progressive or chronological development of the application of biochar and AMF. It started in 1984 and it progresses, okay, up to the present 2020. So these are the researchers that conducted the use of biochar and AMF in applications at crops. And then the last one is the table showing the recent studies reported of the application of the combination naman ng biochar and AMF. And this paper actually cited our paper. So here you can see Agagan et al. So this is our research, no? They also cited our paper. So basically, the use of biochar and AMF started yung combination started in 2015. So it's very recent lang. And this is one of the research development in agriculture, in biological sciences that researchers can further improve. So it progresses up to the present, okay, which is, as you can see, uh, iba't iba yung ginagamit ng mga feedstock. Okay, corn, chicken manure, wood. In our case, we use bamboo. So other studies use reed grass and then cow dung. So you can actually use biomass, manure, wood, leaves, waste products, and so on to convert it into biochar. All right, so I think that's all for my presentation. And before I end, I would like to, uh, you know, extend my gratitude to Cavite State University for, of course, for giving me the opportunity to share this in CSU, Caraga State University. Thank you also for inviting me to talk during this webinar. And of course, this research without it will not be possible without the funding agencies. We have the UST under the ASHRDP, and then we have the USTP card. I was also granted with a thesis dissertation grant, and the A bar because this research is part of the project under the DA bar, and then UP, of course, school and biotech for catering my needs to perform my experiment. With that, I would like to end this presentation and thank you very much for listening.
Thank you very much, Mr. Angelbert Damian Cortes, for the very knowledgeable insights. Surely, there are a lot of interesting things in biological field of science that we'll study from. As a field of science, biology helps us understand the living world and the way species function, evolve, and interact.